where curiosity curls up beside wonder and science takes the scenic route to slumber. Tonight, we're following one of the tiniest travellers in the history of life. It has no heartbeat, no breath, and yet somehow it's one of the most successful entities our planet has ever known. The virus. Silent, invisible, and endlessly strange. Where did it come from? It's a curious thing, really. Life is already a miracle of improbabilities, and yet somewhere along the way, something emerged that isn't quite alive, but not exactly dead either. A creature, a chemical, a mistake with a mission. Viruses don't just inhabit our world, they shape it. They've killed kings, rewritten genomes, and hitchhiked across oceans inside droplets too small to see. And still, their origins are wrapped in mystery. So before we dive into genetic time machines and molecular fossils, let me ask, where are you listening from tonight? Somewhere quiet, I hope. Maybe the stars are out. Maybe it's raining. Maybe you've just set your phone down for the evening and you're letting your mind settle. The way silt settles to the bottom of a slow, still stream. And if this gentle drift into ancient biology helps you sleep, think, or simply breathe a little slower, I'd be quietly grateful if you gave this video a like, maybe even subscribed. It helps this little island of calm find others drifting by. So settle in, slow your thoughts, and let's begin. Because somewhere, between the first flicker of replicating molecules and the rise of the human immune system, lies the story of a creature that isn't a creature at all. It's the story of infection, innovation, and a question that has haunted biology for more than a century. Where did viruses come from? How long have they been with us? And if they predate us, what exactly did they infect? To find an answer, we'll need to travel not just back in time, but back before time meant much to anything with a nervous system. To a world with no flowers, no fish, no feathers, just single cells, sunlight, and the first whispers of competition. But first, we'll start in the present, with a clue hiding in plain sight. Not long ago, nestled deep in the Siberian permafrost, a group of researchers unearthed something odd. It was microscopic, inert, and encased in frozen soil that hadn't thawed in tens of thousands of years. They gently coaxed it back to life, and it woke up. Not a bear, or a beetle, or even a bacterium. A virus. Still infectious, still active, after more than 30,000 years on pause. A kind of molecular ghost with unfinished business. This wasn't science fiction. It was 2014. The virus was nicknamed Pithovirus sibericum, and it didn't infect humans, thankfully, but rather amoebas, those humble blobs that swim through puddles and peat bogs. It was massive for a virus, big enough to see with a light microscope and beautifully odd. Not like anything in our medical textbooks, not like the sleek, brutal elegance of influenza or HIV. This one was boxy, almost clunky. It looked more like a relic than a threat, and in many ways that's exactly what it was. A time capsule, a message from a microbial world long gone, a reminder that viruses have been here not just longer than cities or civilization or even animals, but possibly longer than life as we know it. But if viruses were infecting amoebas in the Ice Age, and amoebas have been around for billions of years, then how far back does this rabbit hole go? How ancient are these invisible agents? And if they're not quite alive, what does it even mean for them to exist? For years, scientists assumed viruses were relatively young, parasites that evolved after cells, opportunists that latched onto the machinery of living organisms and learned to hijack it. But as we peered deeper into genetic history, something strange kept turning up. Viral genes weren't just tucked into viruses, they were everywhere. In bacteria, in plants, in you. Over 8% of the human genome, that's 1 out of every 12 letters in your DNA, is made of viral fossils, remnants of ancient infections, ghosts of pandemics long forgotten. And not just human ghosts. Every species we've looked at, from mushrooms to manatees, carries viral DNA like hidden birthmarks. Even the humble platypus, who already has enough going on, carries entire viral blueprints in its chromosomes. These aren't accidental, they're archaeological clues, so we start piecing it together. And somewhere between Siberian ice and human chromosomes, a hypothesis begins to take shape. Maybe viruses didn't come after life. Maybe they were there at the beginning. Maybe, just maybe, they helped build the world they now infect. And to see how that could be possible, we need to dig a little deeper. Into soil, into stone, into slime. Picture this. The year is about 1977, and a microbiologist named Carl Woese is tinkering with genetic code, comparing RNA sequences in tiny organisms that had barely been studied. What he found wasn't a branch on the tree of life. It was an entirely new domain, archaea, life's forgotten third domain, neither bacteria nor eukaryotes. And just like that, the microbial world tripled in depth overnight. Why does that matter here? Because lurking around these ancient cells were viral forms so strange, so unlike anything in modern medicine, that they might as well have been from another planet. Some had tails like syringes, others like spindle-shaped missiles, and many of them didn't kill their hosts. They just borrowed them, peacefully, parasitically, like sleepy roommates sharing a cell. It turns out the oldest parts of life, the root systems of biology, are crawling with viruses. Not just modern ones, but deep ancestral lineages. Viral families that don't match anything we've sequenced before. Organisms whose evolutionary stories are older than trees, older than fish, older perhaps even than DNA itself. There's a theory, controversial but delicious, that viruses might have been present at the dawn of life, that before there were cells, there were self-replicating bits of RNA, floating in warm little ponds, hijacking chemical gradients and trying, in their own mindless way, to make more of themselves. Because before life as we define it, with cell walls, metabolism, division, there may have been a more primitive phase, something looser, more experimental. The RNA world hypothesis suggests that the earliest proto-life forms weren't cells at all, but molecules that could copy themselves, mutate, and compete. And guess what viruses are excellent at? Copying, mutating, competing. In that primordial chemical soup, it's possible that viruses weren't intruders. They were just another player in the game, perhaps even the first. We think of viruses as destroyers, invaders, saboteurs. But zoom out and you'll see a stranger role emerge, that of genetic tinkerer. Viruses, in their constant attempts to replicate, shuffle genes between hosts. They break things, yes, but sometimes they build. They stitch together new blueprints, they force adaptation, they jumpstart evolution. Like little molecular editors, they cross out old lines and write in new ones, whether you ask them to or not. And sometimes their edits stick. Endogenous retroviruses, for instance, are a type of virus that, millions of years ago, infected the germ cells of animals, sperm or egg, and left a copy of themselves behind. That copy was passed on, and on, and on, until it became part of the species itself. There's a piece of viral DNA in your genome that without it you wouldn't be here. It codes for a protein called Syncytin, which helps build the placenta. Yes, the organ that nourished you before birth was partly made possible by a viral gene. Not an accident, not a one-off, a merger. We tend to think of evolution as a tree, but perhaps it's more like a web, with viruses acting as the knots, tying branches together that would otherwise remain strangers. They are the vectors of surprise, the stowaways who rewrote the map. And so, as we sequence more genomes, dig deeper into permafrost and sift through the fossil record of infection, we begin to see viruses not just as villains, but as historians, ancient messengers, architects. But still and still, one haunting question lingers. If viruses are so old, older than cells maybe, where did they come from? Some researchers argue they began as rogue genes, tiny chunks of genetic material that broke away from full cells and learned to replicate independently. A sort of molecular rebellion. Over time, these genetic fragments became more sophisticated. They developed protein shells, clever delivery systems, and the uncanny ability to hack into hosts. In this view, viruses are like stripped-down parasites, reduced life forms that once were more complete. Others believe the opposite, that viruses predate cells, that they are not degenerates, but forerunners. The RNA world, if it existed, may have been dominated by virus-like entities, molecular machines that could replicate and evolve before cellular life had even begun. In this model, cells didn't come first. They were a late invention, a response to viral pressure, a defense against infection, life, born in fear. And then there's a third idea, that viruses are neither before nor after cells, but something that emerged alongside them. Not from within, not from without, but from between. An emergent property of molecular life, born from the constant exchange of genes and the restless search for replication. A biological side effect that became a global force. No one theory fits neatly. And maybe that's the point. Viruses don't fit. They exist in the grey space between categories. Alive, but not living. Complex, but inert. Architects and arsonists. And all around us they continue their quiet work. In the oceans, viruses kill an estimated 20% of all microbial life every day, recycling nutrients, fueling ecosystems. In the soil, they shape microbial communities that help plants grow. In your body, trillions of viruses, most of them harmless or helpful, form a virome as unique as your fingerprint. They are not invaders from outside life. They are part of it, threaded into our past, whispering through our cells, reminding us in their strange, silent way that even the smallest things can leave the biggest marks. So when did the first virus appear? We don't know. Not exactly. We may never know. Because viruses don't fossilize well. They don't leave bones. They leave impressions in DNA, in disease, in the accelerating story of life. But they were there, in the beginning, or close to it, writing in a language of infection and inheritance. Not monsters, not aliens, just the oldest writers we've ever read. To understand the origins of viruses truly deeply, we need to step back. Not just thousands of years or millions, but billions. Picture a time so ancient that the moon still sat closer in the sky, and Earth spun faster, casting off shorter days. The sun was dimmer, the oceans darker, and the air, well, it wasn't air the way we think of it. There was no oxygen to breathe, no ozone to shield the surface, no wind brushing through forests because there were no forests, no trees, no flowers, no birdsong. The Earth was a water world, cloaked in shallow seas and battered by asteroid rains. Volcanoes hissed through cracks in the crust. Lightning danced across clouds filled with methane and ammonia. And below the waves, hidden from the searing surface, life was beginning to whisper. Not much to look at first. No gills, no teeth, no eyes. Just molecules folding into new shapes. Chemistry pretending to be biology. The first living things, or the first things we would call living, were little more than bubbles of fat holding chains of molecules that could copy themselves. Not even cells yet. Protocells, precursors. In the warmth of underwater vents, or perhaps in the cool puddles left behind by tides and rains, strands of RNA began experimenting with existence. These weren't organisms in the traditional sense. They didn't have metabolism, or membranes, or any sense of self. But they could replicate, they could mutate, they could change. And that, at least for evolution, is enough. This was the so-called RNA world. A hypothetical phase in Earth's history where RNA, not DNA, was the molecule of inheritance. RNA is a remarkable substance. It can carry information like DNA, but it can also fold into shapes that act like enzymes, tiny machines that speed up chemical reactions. In this ancient world, RNA didn't just store the recipe, it also stirred the pot. Now imagine this early, fragile life, suspended in water, tossed about by tides, battered by radiation, vulnerable to every chance disruption. Replication was clumsy, errors were common, but evolution, in its infinite patience, doesn't need perfection. It just needs persistence. And persistence came in many forms. Among the swirling soup of molecules, some forms began to behave differently. They didn't build cell walls. They didn't maintain any of the complex machinery that others were starting to assemble. Instead, they copied themselves by hijacking those who did. These may have been the earliest viruses. They weren't alive, not fully. But they had one powerful advantage: simplicity. While cells had to carry all the tools for replication and maintenance, these protoviruses outsourced everything. They became masters of minimalism. Just enough information to slip inside, take control and escape. That was it. We can't say what they looked like. No fossil remains, no bones, no shells, just a molecular trace in the evolutionary shadows. But the logic of their existence is written into the deepest scripts of biology. Even today, there are viruses that rely entirely on the machinery of their hosts. They bring almost nothing with them. They are messages more than machines. As time marched on, slow, relentless, quiet, those early replicators gave rise to cells, true cells, encased in lipid membranes, with genetic material and basic metabolisms. They formed communities, they traded genes, they began to shape the planet, and viruses followed them, or perhaps in some way led them. By the time Earth was 1 billion years old, simple life had spread across the oceans. Bacteria, archaea, and the earliest hints of what would become complex cells, eukaryotes, were beginning to emerge. The oceans were vast, but life was small. It clung to thermal vents, crept across muddy shallows, floated in colonies beneath the waves. This was a planet ruled by the microscopic. And it still is. Even today, by number, the vast majority of Earth's biomass is microbial. We just don't see it. But among these ancient forms, viruses were already playing a role. They infected, they killed, they swapped genes between species. They introduced mutations that reshaped entire lineages. They carried bits of RNA or DNA across boundaries that biology had barely begun to draw. Imagine the early Earth as a great molecular experiment. Every cell a lab, every tide a shuffle, every virus a wild card. There is a class of viruses today, giant viruses, that still haunt these early patterns. Discovered in the early 2000s, giant viruses are larger than some bacteria. They carry hundreds, sometimes thousands of genes. Some have DNA repair mechanisms, some have translation tools, some scientists suspect they might be remnants of a lost branch of life, a fourth domain. Not just viruses, but something that once lived on its own and was slowly reduced to what we now see, the echoes of ancient independence. And then there are the Virophages. Yes, viruses that infect other viruses. They target giant viruses specifically, hijacking the hijackers. It's a Russian doll of infection. A reminder that even in the viral world, power structures can collapse. These are not signs of simplicity. They are signs of ancient complexity, of ecosystems that have been playing the game of life for far longer than we can fully grasp. Meanwhile, the Earth itself was changing. The oceans were breathing, bacteria were releasing oxygen, and slowly, very slowly, the atmosphere began to transform. This great oxidation event, around 2.4 billion years ago, was a planetary catastrophe and a biological revolution. For anaerobic life, it was toxic. For viruses, it was opportunity. New hosts, new environments, new tools. Eukaryotes, cells with nuclei and internal compartments, began to appear around this time, and as they rose, viruses adapted again. The complexity of the cell gave rise to the complexity of infection. Some viruses evolved envelopes, bits of membrane stolen from their hosts. Others developed precise attachment proteins, lock-and-key mechanisms honed over eons. Every step of biological innovation was met with viral countermeasures, not because viruses think, they don't, but because evolution does. A mindless, tireless engine, always testing, always tweaking. And viruses are its sharpest tools. By the time multicellular life began, sponges, simple worms, colonies of algae, viruses were already old. They had infected every domain of life. They had shaped genomes. They had driven extinction and innovation in equal measure. When the Cambrian explosion bloomed some 540 million years ago, it was a firework of form. Limbs, shells, eyes, nervous systems, all appearing in a geological blink. But beneath the visible changes, the viral world churned on, hidden, pervasive, ancient. Every species that emerged carried viral scars, some inherited through DNA, others etched into behavior, resistance, immunity. The evolution of the immune system itself may owe its sophistication to the pressure of viral infection, natural selection's most persistent antagonist. Even bacteria developed defences. The CRISPR system, which we now use as a gene editing tool, began as a viral memory bank. Bacteria store fragments of viral DNA in their genomes like mugshots. When the same virus tries to invade again, the cell recognises it and cuts it up. Primitive immunity, elegant revenge. In this way, viruses became part of the biosphere's feedback loop. Killers, yes, but also curators, selecting which genes survive, which traits persist, which lineages endure. And so, through mass extinctions and silent recoveries, through ice ages and infernos, through drift and collision and chaos, viruses endured. They are not passengers on the tree of life. They are the wind that bends its branches. Today, in every drop of seawater, there are more viruses than people on Earth. More than 100 million in a single milliliter. They infect marine bacteria by the trillions daily, reshaping ocean chemistry, driving carbon cycles, influencing climate. They are not just historical curiosities. They are active agents in the planetary engine, and yet, for all their power, all their influence, they remain on the edge of definition. They don't metabolize, they don't grow, they don't reproduce on their own, but they evolve, they adapt, they persist. Are they alive? Maybe that's the wrong question. Maybe they are not alive or dead, but other. A parallel strand in biology's braid, a shadow script, an ancient rhythm running just beneath the melody of life. What we know is this. Viruses have been with us since before us had a name, before vertebrates, before limbs, before the very concept of species. They are not aliens, they are not outsiders. They are Earth's oldest storytellers, writing in the genome, whispering through infection, reminding us always that even the smallest things can carry the weight of deep time. The moment we try to pin down the exact trigger, the spark that turned chemistry into virology, it begins to blur. Unlike an asteroid impact or a volcanic winter, the emergence of viruses wasn't one loud bang. It was a gradual uncurling, a slow unfurling of complexity in the shadows of prehistory. But if we tune our minds to those long, deep rhythms, we can begin to sense the outlines of what happened. Somewhere in the sloshing tides of an early Earth, amidst the swirls of amino acids and the first protocells, something changed. A strand of genetic material, maybe RNA, maybe something even more exotic, began doing something strange. It wasn't just copying itself, it was inserting itself into other strands. It wasn't building anything, not really. It was interfering, slipping into the machinery of other molecules, sneaky, subtle, successful. We don't know what this earliest virus looked like, or whether it would meet today's definitions. It might have been just a handful of nucleotides wrapped in a primitive shell, or maybe no shell at all, just bare RNA, drifting through the warm, mineral-rich shallows, waiting for a cell to bump into. But what we do know is this: viruses don't exist in isolation. They require hosts. They co-evolve, and that means they emerge with life, not after it. As cells evolved, so too did the viruses that infected them, and with every innovation in biology, the viral world responded. One major innovation was the development of DNA. In the RNA world, life was fast and sloppy, mutations were frequent, information degraded quickly. But when DNA emerged, sturdier, more stable, it changed the game. Suddenly, life could hold on to complexity. It could build larger genomes, more intricate enzymes, better defense, and viruses, they followed suit. Some researchers believe that DNA itself may have been a viral invention. That's not poetic exaggeration, it's a serious hypothesis. The idea goes like this. Some early viruses, frustrated by RNA's instability, began using modified nucleotides to protect their own genomes. Over time, those changes conferred an advantage. Eventually, the host cells they infected adopted the same strategy. And just like that, DNA-based life was born. It's speculative, yes, but tantalizing. Think about what that means. If true, the very molecule that defines our biology may have been introduced by a virus. Another possible trigger came from energy. Early cells were limited in how much energy they could harness. But then came a revolution. A merger between two ancient microbes that gave rise to the first eukaryotic cells, the ones that make up all complex life. These new cells had mitochondria, internal engines that churned out energy with startling efficiency. And with more energy came more complexity. Longer genomes, more proteins, new vulnerabilities. Viruses had more to exploit, and they did. Some viruses became specialized, others became huge. We find traces of these early viral expansions in the genomes of our mitochondria, in the nuclear DNA of modern eukaryotes, and even in the patterns of gene regulation that control when and how proteins are made. There are entire families of regulatory genes that seem to bear the fingerprints of viral tampering, sequences that look suspiciously like ancient insertion points, tweaked over millions of years until they became part of the system. But there's another wrinkle. Not all viruses are content to jump from host to host. Some bury themselves deep, integrating into the genome like a forgotten tattoo. These are the endogenous viruses, retroviruses specifically, that reverse engineer their RNA into DNA and sneak it into the host's own genetic code. At first, this seems like a bold parasitic trick, but over time, some of these integrations become permanent. They lose the ability to jump out. They get domesticated, tamed, turned into something useful. That's how we got Syncytin, the viral gene that helps build the human placenta. And that's just one example. There are dozens of such co-opted genes in the human genome, doing everything from suppressing tumours to modulating the immune system. Which raises a profound idea. The line between host and virus isn't as clear as we think. In fact, it may not exist at all. Consider transposons, so-called jumping genes that move around within our genome. They were once considered junk DNA. Now we know they play vital roles in evolution, gene expression and genomic diversity. And many of them seem to have viral origins. They behave like mini-viruses, copying and pasting themselves across the genome. Not exactly living, not quite dead, just restless. It's possible that the earliest viruses weren't even external agents. They may have begun as bits of genetic code that learn to move, to duplicate, to escape. Parasites born from within, the genome turning against itself. Or perhaps just evolving inward, folding complexity upon complexity until something entirely new emerged. There's a story that's often told about how viruses infect cells. They land, inject their DNA or RNA, and hijack the host machinery to make more of themselves. It's dramatic, violent, invasive. But there's another story, quieter and no less true, that viruses sneak in, lie low, wait. Some don't kill, some don't even replicate right away. They become latent, dormant, invisible, part of the host. This latent phase isn't failure, it's strategy. It allows viruses to ride out hard times, to wait for better conditions, to avoid immune detection. It also means that hosts can carry viral genomes for years, decades, even lifetimes. And sometimes, that latency becomes inheritance. There are people alive today, perhaps even listening to this, who carry viral DNA from infections that happened millions of years ago, not to them but to their ancestors. A virus infected a cell in a prehistoric mammal, slipped into its DNA, and stayed. Now it rides quietly in your chromosomes, a passenger from a forgotten epidemic. Viruses remember what we forget. They also travel in ways we're only just beginning to understand. Ocean currents, migrating birds, insect vectors, yes, but also pollen, dust, rain. Viruses have been found drifting in the upper atmosphere hundreds of kilometers above the Earth, carried by winds and electrical currents. There may be entire ecosystems of viruses in the sky, influencing weather, climate, even the global carbon cycle. And they don't just influence life, they end it. Mass die-offs in marine algae, viral plagues in bees, outbreaks in corals, bats, amphibians. Everywhere life gathers, viruses follow. They don't mean to destroy. Destruction is just a side effect of replication. The goal, if you can call it that, is only to copy, to persist. And sometimes, persistence requires violence. There are mass extinction events in Earth's history, great die-offs caused by asteroids, volcanism, climate swings. But there are also microbial events, viral events, epidemics that reshaped entire species. We can see their shadows in the fossil record, in the sudden disappearances of once thriving creatures. We see them in the genetics too, bursts of immune gene evolution, signs of ancient arms races, whole species shifting their defences all at once. When the stakes are life and death, even small organisms can leave enormous scars. But here's the thing, viruses don't just drive extinction, they drive adaptation. By culling the weak, they allow the strong to survive. By forcing new immune strategies, they spark innovation. By inserting genes, breaking rules, bending codes, they shake up the status quo. They are entropy agents, disruptors, catalysts. And in that disruption, new life takes shape. We think of evolution as gradual, but in truth, it often comes in fits and starts. Punctuated equilibrium, as paleontologists call it. Long periods of stasis, interrupted by sudden bursts of change, and viruses may be part of the reason why. They are engines of mutation, vectors of chaos. They shake the tree of life and sometimes new branches grow. So if we're looking for the trigger that gave rise to viruses, the spark, maybe it wasn't a single event. Maybe it was a pressure. A constant push and pull between replication and resistance. A dance between structure and subversion. A need to survive, to persist, to spread. Viruses didn't emerge from nothing. They emerged from necessity. They are what happens when information wants to travel but doesn't want to carry a body with it. And so, they shed everything but the message. The virus, in its essence, is a paradox. It is so minimal it teeters on the edge of what we consider life. No lungs, no mouth, no mitochondria. And yet, when it finds a host, it becomes one of the most powerful replicators on Earth. It's a lesson in biological elegance, that sometimes subtraction is its own kind of innovation. And perhaps that's where we find the true mechanism of viral emergence. Not in a single moment, but in a pattern of reduction. A slow stripping away of the non-essential until only the most vital instructions remained. Replicate, hide, spread. Think of a virus not as a creature, but as a message, encoded in molecules, copied across eons, whispered from one cell to another. The earliest ones may not have known what they were doing, not in any conscious sense, but their chemistry carried purpose. They found stability in simplicity, survival in subtraction. They are the ultimate biological minimalists. And yet, within that simplicity, an extraordinary diversity emerged. Today, we know of thousands of viruses, maybe millions more exist, as yet unsequenced, unknown, and each one tells a story of hosts adapted to, of barriers crossed, of species boundaries tested and occasionally broken. Their mechanisms are just as varied. Some viruses burst their way out of cells like raiders fleeing a collapsing fortress. Others slide out gently, cloaked in a piece of the host's own membrane, like spies disappearing into a crowd. Some viruses bring only instructions, a single strand of RNA ready to be copied. Others arrive fully equipped, carrying enzymes, tools, even repair mechanisms. Some infect the nucleus, some linger in the cytoplasm, some lurk in the genome itself. There are viruses that infect bacteria, known as bacteriophages, with tiny legs that grip the surface and a coiled tail that pierces through like a molecular syringe. They inject their DNA with the precision of a nanobot, hijack the bacterial machinery, and use it to make a factory of themselves. And even among these phages there are different strategies. Some go for immediate destruction, the lytic cycle, a fast and furious takeover. Others choose patience, the lysogenic cycle, where viral DNA hides quietly in the host genome, waiting, sometimes for generations, before reactivating. These choices aren't made by minds, but by molecules, and yet they are strategic all the same. It's the cell's behavior that determines the virus's fate, and perhaps the virus in turn shapes the cell's destiny. This is the great feedback loop of biology. In every ecosystem, viruses act as regulators. They control population booms, they redistribute nutrients, they turn over the soil of the microbial world, and in doing so they drive cycles that ripple outward, from the ocean floor to the highest clouds. One example, marine viruses kill off massive blooms of phytoplankton, which releases organic matter back into the water. That matter feeds other microbes, fuels the food web, and affects how carbon is stored in the ocean, which in turn affects climate, all from a microscopic infection. The climate too shapes viruses. Temperature, humidity, ultraviolet light. These aren't just background conditions, they are forces that influence viral stability, mutation rates, and transmission patterns. A virus that thrives in cold Arctic water may fall apart in the heat of the tropics. Another might flourish in the warmth of a bat's body but collapse outside it. This is part of why zoonotic spillover, the jump of viruses from animals to humans, is both rare and dangerous. For a virus to make that leap, it must adapt to a new host's cells, a new immune system, a new internal temperature. Most fail, a few don't, and those few can change everything. Sometimes a mutation is enough. Viruses mutate constantly. Their replication machinery is error-prone, especially RNA viruses, which means that every generation is a little different. This is a weakness and a strength. Many copies are flawed, non-viable, but among the billions produced in a single infection, one may hit upon a combination that spreads more easily, resists immune detection, or crosses a species boundary. That's how pandemics begin, not with intent, with iteration. And again, we see that same ancient theme, change through repetition, evolution by trial and error over time without design, a dance of molecules that occasionally stumbles into power. One of the most remarkable modern insights into viral change comes from metagenomics, the sequencing of genetic material directly from environmental samples. With this technique, scientists have begun discovering thousands of previously unknown viruses, not from patients, not from petri dishes, but from ocean water, soil, sewage, even glacier ice. Many of these viruses have no known relatives. They don't fit into our existing taxonomies. Their genomes are strange, filled with orphan genes, sequences we've never seen before, genes that might be remnants of ancient life, or glimpses of possibilities evolution didn't pursue. In 2018, researchers uncovered a group of viruses in Brazilian amoebas that had almost no recognizable genes at all. They called them yarn viruses because their shape looked like tangled string and their genetics were even more tangled. They didn't resemble any known viral family. It was as if evolution had gone off on its own detour, a separate experiment running in parallel. And it may have been. Some scientists now believe that viruses don't belong on the tree of life at all. They may represent an entirely separate evolutionary domain, not a branch on the tree, but a shadow beside it, a different strategy for surviving time. In this view, viruses aren't failures of complexity, they're masters of efficiency. Think of the genetic material of Earth as a vast library. Most life forms are like books, heavy, self-contained, full of instructions and diagrams and footnotes. Viruses, by contrast, are like anonymous slips of paper tucked into the margins, short, punchy, and often disruptive, but sometimes they say something worth remembering, and occasionally they change the story entirely. We still don't know where the very first virus came from. We may never know. The origin has no fossil, no landmark, only possibilities. Perhaps it arose from an early cell, a rogue piece of RNA that escaped and learned to copy itself. Perhaps it co-evolved with life, a shadow twin from the beginning. Perhaps it was born from the pressures of competition, a stripped-down survivor, built from the leftover scaffolding of a more complex organism. But whatever its origin, the virus, or rather, the viral strategy, proved successful. So successful that it spread across every domain of life, across every ecosystem, across every era. Even now, as we speak, your body is full of viruses. Most are harmless, many are helpful, they shape your immune system, your microbiome, even your brain chemistry. Some scientists believe that viral remnants in our DNA influence how we develop as embryos, how we fight infection, how we age. They are not just visitors, they are part of the architecture, and perhaps that's what makes them so difficult to categorize, because they are in between, not living, not non-living, not entirely outside the body, but not fully of it either. They are liminal things, thresholds in motion, and liminal things are powerful. We see this power in mythology, in folklore, the trickster, the shapeshifter, the creature that lives between worlds. Viruses occupy that same role in biology. They are the tricksters of evolution, the shapeshifters of genetics. They cross boundaries that other life forms cannot. They blur the line between self and other. They rewrite the rules mid-game, and in doing so they remind us that life, real life, is not tidy. It is not symmetrical. It's not always polite. It is messy, experimental, recursive, strange. Viruses are part of that strangeness. They carry the memories of our ancient oceans. They echo the whispers of early RNA. They embody the principle that even the most inert particle can carry transformation, that the smallest things can change the largest systems, and perhaps most beautifully of all, they endure, not in spite of their simplicity, but because of it. So when we ask, where did the first virus come from, we're not just asking about biology, we're asking about beginnings, about how life itself can be infected with possibility, about how the universe in all its chaos found a way to whisper a message and make it stick. We are asking about us, because without viruses we wouldn't be here, not like this, not with our immune systems, our placentas, our molecular scars, our inherited code. We are the sum of our infections, and we are stronger for them. The first virus may be long gone, or it may still be here, circulating quietly, wrapped in a new host, a new name, a new disguise. It may not remember what it once was, but it has never stopped becoming. By the time viruses were well established, and by well established, we mean quietly orchestrating infections across entire microbial worlds, Earth had already undergone a dramatic transformation. Life, once a scattered experiment in shallow water, had begun to organize itself, not into intelligence, not yet, but into ecosystems, interdependent networks of energy, chemistry, and chance. And viruses, ever the invisible puppeteers, were already pulling threads. It's easy to miss because their footprints are so faint. They don't leave fossils, they don't fossilize bones or build towering reefs, but their effects ripple through everything that does. Because when a virus infects, it doesn't just take, it changes. Let's imagine an ancient microbial mat, a living carpet of bacteria and archaea lining the floor of a prehistoric ocean. It looks still, peaceful. But on the molecular level, it's a battlefield. Cells grow, divide, release waste, take in sunlight or chemicals, and compete, always, for space, for nutrients, for survival. Then a virus arrives, just one at first, a tiny strand of genetic ambition. It finds a host, enters, and replicates. And just like that, everything shifts. Infected cells die, bursting like tiny water balloons. Their contents spill out, feeding nearby microbes. Population numbers crash, then recover. New niches open up. Some microbes develop resistance, others mutate. The survivors carry forward not just their own DNA, but the experience of infection, coded into new proteins, new defenses, new behaviors. This is viral ecology, a dance of death and rebirth, feeding the system. In modern oceans, this still happens. Every single day, viruses kill off an estimated 20–40% of marine bacteria. That might sound catastrophic, but it's not. It's essential. Without this culling, microbial blooms would run unchecked, exhaust their environments, and crash far more dramatically. Viral infection recycles nutrients, nitrogen, phosphorus, carbon, back into the system. It keeps life humming at equilibrium. And it's not just the sea. In soil, viruses shape the behavior of fungi, bacteria, and even plant roots. Some viruses help plants survive drought or heat stress. Others suppress or encourage root symbioses. Still others travel between soil and air, part of a little understood aerosol web that links ecosystems across continents. They are, in effect, the planetary editors, but these are the gentle notes. Elsewhere in history, viruses have played louder, darker chords. Take the rise of oxygen, the Great Oxidation event more than 2 billion years ago. As cyanobacteria began pumping oxygen into the atmosphere, they triggered a global crisis. Oxygen then was a toxin. It corroded cellular components, damaged DNA, and disrupted ancient metabolisms. Many anaerobic organisms died. Some fled underground or into deep ocean trenches, but others adapted. And part of that adaptation may have involved viruses. In the chaotic gene swapping that followed this oxygen revolution, viruses acted as couriers. They carried antioxidant genes, protective enzymes, bits of new metabolism from one host to another. They helped write the playbook for surviving in an oxygenated world. It's a pattern that would repeat. Whenever life stumbled upon a new invention, photosynthesis, multicellularity, internal skeletons, viruses were there, reshuffling the deck, testing combinations, seeding risk. Some of those risks became revolutions. When eukaryotes emerged, those complex cells with nuclei and mitochondria, they brought with them vulnerabilities, larger genomes, slower replication, more machinery to sabotage. For viruses, this was a golden age. New hosts, new niches, new strategies. And so the viruses diversified too. Some learned to cloak themselves in host membranes, enveloped viruses, which made them harder to detect. Others evolved capsid proteins that match cellular receptors with eerie precision. Still others developed latency, the ability to remain hidden in the host's genome for years, even decades before reactivating. Latency changed everything. No longer did a virus have to kill its host immediately. It could wait, bide its time, spread silently, become, in some sense, part of the host. This gave rise to an arms race, one that's still going. Hosts developed immune systems. First, simple barriers, cell membranes, enzymes, pattern recognition. Later, In multicellular organisms, adaptive immunity, antibodies, T cells, memory responses. But for every defense, a counter-defense evolved. Viruses mutated their surface proteins, mimicked host molecules, disrupted immune signaling. Some went further still. Certain viruses, like Epstein-Barr or cytomegalovirus, encode proteins that actively manipulate the host immune system. They turn off detection pathways. They suppress apoptosis, programmed cell death, so that infected cells linger longer, like guests who won't leave the party. And even this had consequences. Long-term viral infections may have driven the evolution of cellular aging, senescence, and the tight regulation of cell division. In effect, viruses may have helped shape the very rhythms of multicellular life. Then came the leap to tissues, organs, bodies. Multicellularity was a grand experiment, one that allowed for specialization, complexity, and eventually sentience. But it also meant new vulnerabilities. A virus no longer had to kill the whole organism, just a part of it. The lungs, the liver, the blood. An infection could spread from cell to cell, tissue to tissue, sometimes with astonishing speed. And that's where pandemics begin. We think of pandemics as modern horrors tied to cities, trade, air travel. But the mechanisms are ancient. The sudden, explosive spread of a virus through a population is as old as herds, flocks, schools of fish, as old as community. And the aftermath is often more than biological. A viral outbreak doesn't just change the infected, it changes the survivors. In populations exposed to repeated viral stress, we see immune systems becoming more robust, more varied. We see genetic bottlenecks where only the resistant survive. We see behaviours shift, more grooming, less contact, even changes in mating. These aren't conscious decisions, they're evolutionary feedback, and viruses keep playing both sides. Some viruses form lifelong relationships with their hosts, symbioses that blur the line between infection and partnership. The polydanaviruses that help parasitic wasps suppress their host's immune system. The viruses that enable heat tolerance in certain grasses. Even bacteriophages that defend their bacterial hosts from other phages. Not all viruses are destructive. Some are guardians, some are engineers. And yet there are times when the viral hand hits harder. When retroviruses invade the germline, the DNA of sperm or egg, their genetic cargo becomes permanent, passed down, inherited, a genomic stowaway. And sometimes those viral insertions are benign. Other times they become regulatory switches, turning genes on or off, sometimes in ways that matter. One such insertion helped mammals evolve the placenta. Another may have shaped the human brain. That's not hyperbole. Some researchers believe that endogenous retroviruses influenced the development of the neocortex, the layered brain structure responsible for language, reasoning, and conscious thought. Viral elements appear in genes that regulate neural development, synapse formation, even plasticity. It's a strange thing to consider, that the scaffolding of human intelligence might carry the fingerprints of ancient infections, and stranger still, that the same forces can unravel. Some modern diseases may be echoes of these ancient viral encounters, autoimmune conditions, cancers, even some neurological disorders. When viral genes misfire or reawaken, the results can be unpredictable. This is the viral double-edged sword. They bring change, sometimes beautiful, sometimes brutal, but always transformative. The Earth remembers. The Earth remembers even when we don't. Not in diaries or monuments, but in the layered silt of ocean floors, in the ice cores of forgotten winters, in the code threaded through chromosomes. For every extinction, every adaptation, every strange left turn in the story of life, there are viral fingerprints smudged in the margins. Quiet, patient, permanent. Take, for instance, the long shadow of viral infections on reproduction itself. It's not just the placenta, it's the whole choreography of pregnancy, from immune tolerance to nutrient transfer. In most mammals, the mother's body must suppress its own defense systems to avoid attacking the fetus, which is genetically speaking half foreign. And some of the immune modulating genes involved in that truce, they're viral in origin, gifts from retroviruses, once intruders, now midwives. The same applies to developmental switches, when certain genes turn on or off, how early embryos divide, what axis the body plans itself around. Again and again, we find viral elements acting as regulatory sequences, like old bookmarks left in a very strange instruction manual. But the virus's reach doesn't end in the womb, it continues through life. In our immune systems, for example, we find a complex ballet of pattern recognition, proteins that scan for invaders, memory cells that recall past battles, mechanisms for distinguishing friend from foe. This machinery is ancient and much of it seems to have evolved under viral pressure. Even CRISPR, the gene editing tool revolutionizing modern medicine, began as a microbial memory system, a way for bacteria to remember and retaliate against phages. The idea that our most intimate defences, the code of our immune cells, was sculpted by ancient attackers feels almost mythic, like learning that your house was designed by the thief who once tried to break in, and then broke in again and again, until you rebuilt the locks around him. But it's not just about humans. Every organism with a genome, from fungi to ferns to whales, shows traces of viral warfare. In plants, viruses have driven the evolution of chemical defences, bitter saps, reactive compounds, even structures that discourage viral vectors like aphids or beetles. Some plants produce antiviral compounds so potent they've inspired human medicine: quinine, nicotine, salicylic acid. Meanwhile, insects, carriers of countless viruses, have evolved their own form of RNA interference, a molecular trick to silence viral genes before they spread. In fact, some insects have co-opted entire viral genomes to bolster their own immune arsenals. Nature, it seems, is not above stealing from its enemies. Even social behaviours may bear the subtle influence of viral pressure. Animals that live in tight-knit groups are more susceptible to outbreaks, and over time, they evolve norms that mitigate the threat. Grooming, scent checking, even territorial spacing can all serve epidemiological functions. Among primates, some researchers believe that certain social structures, who sleeps where, who touches whom, who eats first, are shaped as much by immune strategy as by dominance. And in humans, it's tempting to wonder. We wash, we isolate when sick, we develop taboos around contamination. We learn, generation by generation, to fear the unseen, to pass down warnings, to tell stories about plagues, punishments, invisible threats from the air. Myth becomes memory. Memory becomes reflex. And even today, the idea of infection shapes our politics, our economies, our digital metaphors, computer virus, viral marketing, quarantine zones, firewalls. We speak the language of virology, even when we're not talking about biology. But what about the darker aftermaths? The wounds left by viral events we didn't survive. Not every organism adapts. Not every ecosystem recovers. There are extinction events written not in asteroid dust or lava flow, but in the silence left behind when a virus jumps into a new host and spreads unchecked. We've seen it happen. In the chytrid fungus devastating amphibians, in the hemorrhagic fevers collapsing bat populations, in the pox that once swept through indigenous American communities after contact with European colonists, bringing with it viral hitchhikers to which they had no resistance. Viruses don't care who you are, but they do care how many of you there are, how closely you live together, how often you share breath. They exploit connection, and as life grew more connected, from cells to colonies to continents, viruses rode those links like bridges. Sometimes the effect is immediate. A population crashes, a food web collapses, predators vanish, prey overshoots, balance resets. Other times the impact is delayed, a genetic change that leads to a new trait, a new behaviour, a new branch on the tree of life. a mutation introduced by a viral misfire that, 1,000,000 years later, becomes a gene for vision, or feathers, or milk. These changes are invisible in the moment, but they accumulate, and over time they shape entire lineages. It's no exaggeration to say that without viruses, evolution would be slower, less experimental, less weird. Because viruses aren't bound by the same rules. They don't inherit linearly. They jump, they recombine, they cut and paste and shuffle. They take bits of one genome and drop them into another. This process, called horizontal gene transfer, is wildly destabilizing and wildly creative. It's like evolution with a hacksaw, and sometimes that's what life needs. Of course, there's a price. Always. The same viruses that spark innovation can also trigger collapse. The same mobile genetic elements that grant a survival edge can, when misregulated, cause cancer. The same retroviruses that helped us evolve placentas also help us build tumours when things go wrong. It's a reminder that biology is rarely clean. It's a constant negotiation, a fragile truce, and viruses sit at the centre of that tension. There is a poetic irony here. For centuries, we thought of viruses as enemies, unnatural, intrusive, the very opposite of life. But the deeper we look, the more we realise that they are woven into life, that they've been here since the beginning, that their chaos helped make our order. Even now, in cutting-edge research, we use viruses to cure disease. Modified viruses carry corrected genes into cells for gene therapy. Viral vectors are used in vaccines. The same machinery that once hijacked our bodies is now being repurposed to save them. We have, in a sense, made peace with our oldest invaders, but perhaps it's more accurate to say we've become them in part. Your genome is a palimpsest, a manuscript written over 1,000 times, with each generation adding, deleting, scribbling in the margins. Viruses have been among the most prolific authors. They don't just infect, they edit. And while we may never know what the very first virus looked like, whether it was a rogue RNA strand, a molecular mistake, or a spark from some forgotten ancestor, we can still feel its impact in our cells, in our history, in the very architecture of the world around us. They were here before we named them. They will be here long after we forget. And their story, like ours, is still being written. The Earth has never stood still, not in its plates or in its climate, not in its creatures or its chemistry. And while we often think of life evolving in a gentle arc, from microbes to mammals, from sea to sky, the truth is that Earth's biological story has been one of repeated collapse, sudden stops, great undoings, mass extinctions, moments when the diversity of life plummeted, when the oceans went silent, when forests blackened, when entire evolutionary experiments were wiped from the board. And yet, in the ashes of these events, life returned, changed, slimmer sometimes, hungrier, but always ready to begin again. We tend to focus on the dramatic causes, the asteroid that ended the reign of the dinosaurs, the volcanic hellscapes that poisoned the seas during the Great Dying, the Snowball Earth events that froze the planet to its fingertips. But between these loud chapters, there were quieter ones, microbial shifts, atmospheric tipping points, slow diebacks, quiet extinctions, and among those subtle pressures, viruses may have played roles we're only just beginning to recognise. To understand how, we must step back, farther than we're used to, beyond the dinosaurs, beyond the forests, beyond even the Cambrian explosion. Back to the Precambrian world, a planet still learning how to host complex life. At this time, life was largely microbial. Bacteria, archaea, and the earliest eukaryotes swam in oceans rich in metals, poor in oxygen, and constantly stirred by tectonic unrest. These microbes built stromatolites, layered mats of cyanobacteria that still exist today in some shallow coastal areas. They breathed out oxygen as waste, changing the very air itself. And then, about 2.4 billion years ago, that oxygen accumulated enough to trigger what we now call the Great Oxidation event. For many organisms, it was a catastrophe. Oxygen is a reactive, corrosive molecule. It damages proteins. It shreds DNA. It was, to ancient anaerobic life, a poison. Some scientists believe that viruses help speed up this collapse, acting as agents of stress, targeting weakened cells, accelerating the breakdown of populations that couldn't adapt. And as survivors emerged with better defences, antioxidant enzymes, DNA repair mechanisms, the first inklings of mitochondria, viruses followed them too, shifting their targets, updating their tactics. What came next was one of Earth's great pauses, a long, low plateau in biological complexity. For nearly 1 billion years after the Great Oxidation, not much seemed to happen. It's a period paleontologists sometimes call the boring billion. Few fossils, little oxygen fluctuation, a world in stasis. But beneath the surface, things were stirring, and viruses, always quiet, always watching, were among the agents of change. The earliest eukaryotes were beginning to diversify. Single-celled protists were experimenting with new ways of moving, feeding, mating. Some began to engulf other cells, a behaviour that would eventually give rise to mitochondria and chloroplasts. Others began to develop internal skeletons, primitive sensory structures, even simple sexual cycles. Viruses infiltrated all of it. Their genomes were found in nuclear DNA, in organelle DNA, in plasmids and transposons, and with every infection they shuffled the deck, introducing mutations, moving genes from one species to another, sometimes causing cancer-like growths that paradoxically led to more complex cell behaviours. They were, in many ways, the chaos that drove creativity. And then, the Earth froze. The Snowball Earth hypothesis suggests that around 720 and again, around 635 million years ago, the planet underwent extreme glaciations. The oceans covered in ice from pole to equator, temperatures plunging, sunlight bouncing off white deserts of frost. Life, again, was pushed to the edge. Where did it survive? Maybe in hot springs, maybe under thin ice near the equator, maybe in hydrothermal vents deep in the ocean. And again, viruses would have been there, not just as killers, but as curators, pruning the weak, maintaining population balance, perhaps even helping cells swap genes for cold resistance, for antifreeze proteins, for tighter membranes. Because when the ice retreated, when the world thawed and the ocean stirred and sunlight poured back into the sea, something changed. Multicellular life bloomed. Within a few million years, we see signs of animals, soft-body things, flat frond-like organisms, crawlers, filter feeders, and just after that, bang, the Cambrian explosion. In evolutionary terms, it was a supernova. In a geological blink, 20, maybe 30 million years, nearly every major animal body plan appeared. Vertebrates, arthropods, mollusks, an arms race of anatomy. Why did it happen? No single answer satisfies. Maybe it was rising oxygen levels. Maybe ecological complexity reached a critical threshold. Maybe it was the development of predation, the first jaws, the first exoskeletons, the first blood. But some suggest that viruses helped prime the world for this leap. By shaping gene regulation, by encouraging horizontal gene transfer, by acting as mobile elements that allowed early animals to adapt faster, change quicker, rewrite themselves more freely. If natural selection is the editor of life, viruses are the rogue poets scribbling in the margins, and the scribbling didn't stop. Throughout the Paleozoic and Mesozoic eras, as life colonized land, grew trees, evolved wings and wombs and war cries, viruses kept pace. When the first forests rose, giant ferns and towering lycophytes, plant viruses likely travelled with insects, fungi and airborne dust. Some may have helped plants develop resistance to drought or salt. Others may have caused diebacks, setting the stage for ecological turnover. When amphibians crawled onto land, when reptiles learned to lay eggs, when mammals suckled their young, viral DNA was there, hiding in the background, tweaking hormone regulation, immune tolerance, sensory perception. The fossil record speaks mostly in bones and shells, but the genetic record whispers louder. It tells us that innovation often comes from conflict, that creativity can be provoked, that life doesn't just adapt to viruses, it adapts through them, and sometimes it breaks. The Permian extinction 252 million years ago was the worst the planet has ever seen. Over 90% of marine species gone, forests collapsing, the biosphere unravelling. The primary cause? Massive volcanism in what is now Siberia, fire, ash, CO2, methane, climate spiral. But some researchers believe that secondary stressors, including microbial and viral outbreaks, may have contributed to the collapse. When ecosystems teeter, pathogens surge, population density plummets, but disease dynamics intensify. Survivors are often those who can resist infection as well as starvation or heat. And in the long shadow of extinction, viruses once again helped write the recovery. Because with the rise of the survivors, the ancestors of modern mammals, reptiles and early dinosaurs, came new opportunities, empty niches, unclaimed territory. Evolution took wild leaps, and viruses came along for the ride. By the time birds took flight and mammals found fur and early primates swung through trees, the virome, the entire collective viral population of the biosphere, had become staggeringly complex. Each lineage carried its own viral passengers, some ancient, some newly acquired, some stowaways, some saboteurs, but all of them part of the system. We tend to think of time as linear, a long arrow pointing forward, progress stacking neatly on progress. But evolution doesn't move like that. It spirals, loops, retraces its steps, loses what it once built, then rediscovers it in a different form. And viruses, more than any other biological force, expose these loops. They don't just infect a species once, they return in new shapes with familiar tricks. A virus that plagued mammals 50 million years ago might not be gone, only sleeping, buried in our genomes, waiting to be reawakened by mutation, by chance, or by a shift in the environmental balance. And those awakenings leave marks. Consider the ancient battle between retroviruses and vertebrate genomes. Over millions of years, retroviruses have infected the germline, passing from parent to offspring like cursed heirlooms. At first, the host fights back. Genes mutate to silence the intruder. New proteins evolve to cut up viral RNA. But sometimes the virus wins, or at least negotiates a truce. Eventually, the viral sequence no longer causes disease. It just stays, forgotten, harmless. And then, over time, something stranger happens. The host starts to use it. What began as a parasite becomes a partner, a code fragment repurposed, a viral protein transformed into a tool for building, for regulating, for sensing the world. These transformations are not rare. In fact, they may be essential. Endogenous viral elements, ERVs, make up roughly 8% of the human genome. That's more than all the genes that actually code for proteins. The instructions for making you are outnumbered by the ghosts of ancient viruses. Some of these ghosts still function. Others are fragments, like the bones of extinct species scattered in a dry canyon. Together, they form a genetic palimpsest, a record of infection and adaptation so vast we're only beginning to decipher it. And this is not just a human story. In whales, in insects, in ferns and fungi, we see the same viral footprints, entire viral families that seem to have passed from one kingdom to another, not by descent but by collision. There are sequences shared between plants and insects that suggest a long history of cross-kingdom viral exchange. There are bacteriophages, viruses that infect bacteria, that carry genes borrowed from animals. The lines blur. The tree of life, once imagined as clean and branching, becomes a tangled braid, and still the pattern holds: crisis, collapse, invasion, adaptation, innovation, then again, crisis, collapse, invasion. And always the virus moves through it all, not as a villain, not as a hero, but as an accelerant. Let's fast forward, past the ice ages, past the Neolithic revolutions, past the first cities and ships and empires, into history, into memory, into the modern age. We've recorded viruses now, named them, sequenced them. We know their shapes, their vectors, their tricks. We map their genomes like cartographers once mapped new continents, with wonder and terror. But the patterns have not changed. When urban density rises, so does the risk of outbreaks. When humans push into new ecosystems, spillover events occur. When animals are kept in close quarters, viral evolution speeds up. We've seen it again and again. In influenza jumping from birds to pigs to people. In Ebola leaping from bats to primates. In HIV crossing species lines and reshaping the modern world. In coronaviruses spreading from wildlife markets into cities, then into headlines, then into the bloodstream of history itself. And each time the question returns, are we prepared? Not just with vaccines and ventilators, but with understanding. Do we see the pattern? Do we remember the loops? Because viruses are not just ancient forces, they are modern mirrors. They show us how interconnected we are, how fragile, how dependent on systems we cannot see, microbial, ecological, genetic. They remind us that life is never isolated, that what infects one species can, with time and luck and mutation, infect another, that the boundaries between them and us are thinner than we'd like to believe, and they remind us too of the power of adaptation. The Black Death reshaped Europe, not only demographically but socially, economically, culturally. Survivors carried forward new ideas about labour, health, mortality. In the wake of HIV, we saw massive shifts in public health infrastructure, medical research, even global cooperation. And yet, we forget. Again and again. History's viral loops do not guarantee learning, only the chance for it. And here, perhaps, is the deepest echo of all. Viruses are not just biological phenomena, they are philosophical ones. They ask hard questions about identity, permanence, change. They force us to confront what it means to be alive, to change, to merge, to carry parts of others inside ourselves. They blur the line between self and other, between foreign and native. After all, when you are made of billions of cells, each one shaped by ancient infections, where do you end and your history begin? Maybe viruses are the price of complexity. Or maybe they're the whisper that complexity was never the goal, only persistence. And if we listen to that whisper, we hear something else too. Humility. The Earth has been host to viruses for billions of years. It will host them for billions more. We are not their targets. We are simply their current terrain. And like wind or fire or time, they will move on when the conditions change. But while they are here, we can learn from them about speed, about resilience, about transformation through disruption. We can see in them the strange beauty of a world that edits itself, that folds old mistakes into new solutions, that carries its ancient scars not as burdens but as blueprints. So the next time we ask, where did the first virus come from? We might also ask, what did it become? And the answer, quietly, overwhelmingly, is everything. The virus is not just in the cell, it is in the structure of life itself, in our ability to adapt, in our capacity to remember, in the very idea that we are part of something older, weirder, more tangled than we will ever fully understand. Because viruses didn't come from somewhere else. They came from here, from Earth, from life's own restless chemistry, and in a way they never left. The story of viruses has always been a quiet one. They don't make noise, They don't cast shadows. They move invisibly, replicate silently, and vanish without a trace unless you know exactly what to look for. So it's fitting, perhaps, that the people who uncovered them were often quiet too. Careful, curious, persistent. The first true glimpse of a virus came not from a microscope, but from a filter. The year was 1892, and a Russian botanist named Dmitry Ivanovsky was investigating a strange disease afflicting tobacco plants. Leaves mottled, growth stunted, farmers confused. It looked like a bacterial infection, but when Ivanovsky passed a sap from the infected plants through a porcelain filter designed to trap bacteria, the disease passed right through. Whatever was causing it was smaller than bacteria, invisible, resistant, and yet still alive, or something like it. 6 years later, Dutch microbiologist Martinus Beijerinck picked up the trail. He confirmed Ivanovsky's results and gave the agent a name, Contagium vivum fluidum, the contagious living fluid. He didn't know what it was, but he knew what it wasn't. It wasn't a toxin, it wasn't a cell, it was something new, something that could slip through the nets of science. And that's where the mystery held. For decades, scientists couldn't see viruses. They couldn't grow them in lab dishes. They couldn't stain them, slice them, isolate them. They could only observe the havoc they left behind. Wilted plants, dying animals, feverish children, shadows without bodies. Then came the electron microscope. In the 1930s, advances in imaging allowed researchers to finally see viruses, to glimpse their shapes, their symmetry, their alien beauty. The tobacco mosaic virus was the first ever visualized, a rod-shaped spiral of protein and RNA. It looked almost elegant, like a machine built by nature at the edge of life. That first image was more than a breakthrough. It was a revolution. Suddenly, the invisible had a face, and once the world of viruses had formed, the floodgates opened. Scientists began cataloguing them. Bacteriophages with their lunar lander legs and tiny syringes, spherical flu viruses with spiky proteins, complex herpes viruses hiding like pearls inside icosahedral shells. Each new virus was like a message in a bottle, floating from a world we'd never seen but had always lived in. And in those early decades, each discovery came with a wave of both fear and fascination. The 1918 influenza pandemic, which had ripped across the globe, killing more people than World War I, was suspected to be viral, though the culprit wasn't confirmed until much later. In its wake, researchers doubled down. Labs were built, institutes founded. Virology, once a footnote in microbiology, became its own frontier. One of the pioneers was Max Delbruck, a physicist who turned to biology because he believed life could be decoded like math. In the 1940s, he and his colleagues founded the phage group, using bacteriophages, viruses that infect bacteria, as model systems to understand the fundamentals of genetics. It was with phages that scientists first proved that DNA, not protein, was the molecule of inheritance. It was with phages that they learned how genes replicate, mutate, recombine. In a sense, modern genetics was born in the shadow of a virus. And then there was Rosalind Franklin, whose X-ray crystallography helped reveal the double helix structure of DNA. Less known is that in her final years, she turned her attention to viruses. She mapped the structure of the tobacco mosaic virus with the same precision she brought to DNA. Her notes brim with admiration for its geometry, its ordered repetition, its molecular elegance. She died before she could finish the work, but others followed. The second half of the 20th century was a golden age for virology, not just in the lab but in the field. Virologists began to travel, to jungles, to deserts, to rainforests and remote islands, chasing outbreaks, collecting samples, tracking disease at its source. In the Congo in the 1950s, researchers began studying hemorrhagic fevers. They didn't yet know they were looking at viruses. just that people were dying in patterns that made no sense. It would take decades to isolate Ebola, Marburg, and their terrifying kin, and longer still to understand their reservoirs, their vectors, their evolutionary origins. Some researchers didn't make it back. The work was dangerous, and yet they returned again and again, not for fame, there wasn't much of that, but for knowledge, for prevention, for the belief that understanding was the first step to protection. Then came the 1980s, and with them HIV. At first it was a whisper, a strange pneumonia in otherwise healthy young men, a cancer rarely seen outside of the elderly suddenly spreading in urban centres. Doctors were confused, concerned, and then overwhelmed. The virus moved silently, slowly, sometimes taking years to manifest symptoms. It targeted the immune system itself, dismantling the body's defences. It had no cure, it spread through blood, sex, and from mother to child. And it killed. The global scientific response to HIV was massive and slow. The virus was identified in 1983 by a French team led by Luc Montagnier and Françoise Barré-Sinoussi. American researchers followed close behind. Within a year, the viral structure was mapped. Tests developed, drugs trialled. But stigma slowed the science. Misinformation flourished. Communities most affected, LGBTQ+, black, poor, were often ignored or vilified. It was one of virology's greatest challenges, not just biologically but morally, and yet the progress was staggering. By the mid-1990s, combination antiretroviral therapy had transformed HIV from a death sentence into a chronic condition. Researchers learned not only how the virus replicated, but how it entered cells, how it integrated into the genome, how it avoided detection. In unlocking HIV's secrets, we learned more about immunology, cell signaling and human genetics than we ever had before, and we built systems, surveillance networks, global databases, rapid response labs, a new kind of virology, global, connected, interdisciplinary. These systems would prove vital when, in the 21st century, viruses began to spill over faster. SARS in 2002, H1N1 in 2009, MERS in 2012, Ebola again in 2014, Zika in 2015. Each time researchers mobilized faster, sequencing happened within days, vaccines were designed in weeks, international cooperation became the norm, even if political will sometimes lagged behind, and then came 2020. COVID-19 wasn't our first pandemic, but it was the first truly global one in the digital age, tracked in real time, debated on every screen, studied by teams across borders. The SARS-CoV-2 virus was sequenced within weeks of its emergence. Vaccines were developed and deployed at record speed. Never in human history had we responded to a virus with such urgency, coordination, and scientific power. But the science didn't stand alone. Behind every genome map, every antibody test, every paper published at 3 a.m. by a sleep-deprived researcher, was a person, a team, a story. Scientists working in BSL-4 labs, fully sealed in protective suits, studying live viruses so deadly they could kill with a pinprick. Field epidemiologists chasing outbreaks in sweltering villages, collecting samples, interviewing families, often without fanfare or funding. Computational biologists modelling viral spread from laptops in kitchen corners during lockdown, and behind them, the ghosts of those who came before, Ivanovsky, Bayerink, Franklin, Delbruck, and the unnamed ones, researchers lost to infection, to burnout, to bureaucratic resistance. Virology is not a discipline built on eureka moments. It's built on patience, on repeated experiments, on long hours and low light and the willingness to be wrong again and again before something clicks. It's also built on fear, on grief, on urgency. Viruses don't wait. And yet, what's remarkable is that so many scientists keep going, not because the work is safe or glamorous or easy, but because they believe the story matters. Because even now there are viruses we haven't discovered, entire families lurking in rainforest canopies, in permafrost, in the guts of deep-sea fish. There are patterns we haven't traced, spillovers we haven't predicted, mysteries we haven't unwrapped, and the only way forward is through curiosity. The same curiosity that led a botanist to test a sick plant in 1892. The same curiosity that built vaccines in record time. The same curiosity that now sends drones and biosamplers into the clouds to study airborne viromes we didn't even know existed a decade ago. Virology is a human story, messy, noble, restless, and it's far from over. Virology, like the viruses it studies, has always existed at the edges, not quite mainstream, not quite settled. Tucked between microbiology and immunology, between medicine and molecular biology. Too small to be seen, too strange to categorize. It was, for a long time, the science of shadows, of whispers, of anomalies that didn't fit the rules. And so, it attracted a certain kind of mind. People who were comfortable in ambiguity, who weren't deterred by the absence of evidence, or the absence of applause. People who saw a pattern in the noise and stayed with it, even when no one else did. One such mind was June Almeida. Born in Glasgow in 1930, she left school at 16 and became a lab technician, examining tissue samples under early electron microscopes. She had no university degree, no formal scientific accolades, but what she had was vision, and not just literal. She had an intuition for structure, a feel for pattern. She developed a technique called immune electron microscopy, using antibodies to clump viruses and make them visible under high magnification. She used it to photograph rubella for the first time, and then, in 1966, she helped image a new type of virus, one with a crown-like halo of surface proteins. She called it a coronavirus, from the Latin corona, meaning crown. The name stuck. At the time, few noticed. She quietly returned to her work, publishing, teaching, training the next generation. It wasn't until the SARS outbreak of 2003, and especially the COVID pandemic of 2020, that her contributions were widely recognised. Her archived images were revisited, her methods validated, her insights celebrated, finally, decades later. Science, like viruses, is often slow to show its full effect, and this is not rare. Many of the most pivotal virologists remain unknown outside their circles. Women and researchers of colour in particular were often written out of the stories they helped build, like Yvonne Barr, who helped co-discover the Epstein-Barr virus, or Dr. Ruth Bishop, who played a key role in identifying rotavirus, a major cause of infant mortality, or Dr. Flossie Wong-Stahl, who first cloned HIV and mapped its genes, paving the way for diagnostic tests and therapies. These scientists didn't just identify pathogens, they helped decipher the inner grammar of infection. They figured out how viruses attach to cells, how they enter, how they hijack. They discovered the receptors, the inhibitors, the resistance genes. They laid the foundations for vaccines, antivirals, public health interventions, and sometimes they found things they weren't even looking for. Because viral research often leads into unexpected corners of science. In studying how viruses insert themselves into host genomes, we learned how to use viruses as gene therapy vectors. In tracking ancient viral remnants in DNA, we uncovered new tools for dating evolutionary events. In building vaccines, we learned how to train the immune system, not just to fight, but to remember. And in chasing viral evolution, we glimpsed the broader logic of life. Take the story of bacteriophages. When researchers started mapping phage genomes, they found an astonishing variety of genes, many with no known function, many more with origins that couldn't be traced to any cellular life. Some seemed to be patchworks, one section from a eukaryotic host, another from a bacterium, another from an unknown virus. It was as if these phages were curators of genetic scrap, collecting bits from across domains of life and stitching them into something that worked. One such discovery, made in the 2000s, would change the field of genetics. the CRISPR-Cas system. Originally observed in bacteria as strange repeating sequences, CRISPR was eventually understood to be a viral memory system, a way for bacteria to store fragments of phage DNA and use them to recognize and destroy future invaders. In other words, an immune system for microbes. This mechanism, ancient, precise, elegant, would be transformed into a gene-editing tool of staggering power. Scientists now use CRISPR to modify DNA in plants, animals and humans with unprecedented accuracy, all thanks to an arms race between bacteria and viruses that's been going on for billions of years. This is the recursive beauty of viral science. The problem often contains the solution. Viruses, once feared as pure destruction, have become tools, teachers, templates for molecular machines. In some cases, they've even become cures. Gene therapy trials now use modified lentiviruses, cousins of HIV, to deliver therapeutic genes into patients with inherited diseases. Oncolytic viruses are being engineered to target and destroy cancer cells, sparing healthy tissue. Viral vectors are used in vaccines for Ebola, COVID-19, and other emerging threats. But every breakthrough comes with caution. The scientists who build these tools are acutely aware of their risks. Viruses mutate, recombine, spill over. What heals today could harm tomorrow, if not handled with care. And so modern virologists carry a heavy responsibility, not just to discover, but to anticipate, to predict viral emergence, to monitor hotspots, to build early warning systems. In this work, field scientists are critical. They're the ones who travel to remote caves to collect bat samples, who work with local communities to track zoonotic spillovers, who study migratory birds, mosquito populations, permafrost cores, sewage samples. They build viral maps the way old explorers charted continents, with notebooks, vials, microscopes and boots caked in mud. These scientists often operate on small grants, under intense conditions, but their work saves lives. Sometimes the discovery isn't the virus itself, but the absence of it. The understanding that a mutation didn't happen, a spillover didn't occur, a chain of transmission was quietly broken. Prevention doesn't make headlines, but it makes history. And woven through all of this is a very human question. Why? Why do they do it? Why spend years chasing invisible threats through data and dust, knowing that funding is precarious, that recognition may never come, that the next pandemic might still outrun them? The answer is simple and profound. Because someone has to. Because knowledge matters. Because even when the story is quiet, the stakes are not. Because every time we understand a virus more deeply, we understand ourselves, our vulnerabilities, our resilience, our interdependence. Because viruses are not just about illness. They're about information, about systems, about adaptation under pressure. They are the language of change spoken fluently in every ecosystem, every lineage, every era. And the scientists who listen, truly listen, are translators of that language. They help us hear what life has been trying to say all along, that we are never alone in our biology, that everything we call individual is stitched together from past interactions, that survival is not about strength but responsiveness, not about purity but exchange. In a world where isolation is easy to believe in, politically, socially, even biologically, viruses tell a different story, one of connectedness, of permeability, of constant recursive transformation. And the scientists who chase viruses into glaciers, jungles, genomes, they are in a way following a trail of mirrors. Each discovery reflects back a new facet of what it means to be alive, and so the work continues, in labs and in libraries, in bat caves and blood banks, in code and culture and conversation. Quiet, patient, unending. A science for shadows, a science for sleep, a science, just possibly, for everything. To understand the full reach of viruses, you must first abandon the idea that life begins and ends with cells. Because viruses do not follow the rules of life, They whisper around them. They are the ghosts in the machine, the punctuation marks in the sentence, the editors who never sleep. So let us begin at their beginning, not the first virus, for we do not know when that was, nor may we ever, but the beginnings we can trace, the chemical architecture, the molecular choreography, the tiny moving parts that allow a virus to be something more than just stray genetic debris. At its core, a virus is deceptively simple. Most consist of only two essential components: genetic material, either RNA or DNA, and a protective coat made of proteins, known as a capsid. Some viruses add a third layer, a lipid envelope, often stolen from the host's own cell membrane, which grants them stealth, flexibility, and peculiar vulnerability to something as mundane as soap. That's it. No ribosomes, no metabolism, no cellular machinery. A virus cannot generate energy. It cannot reproduce on its own. It carries only instructions, and it must hijack a living cell to execute them. But within that simplicity lies a stunning variety. There are helical viruses spiraled like tiny springs, icosahedral ones, geometric and crystalline, complex viruses that look like lunar landers, complete with tails and fibres for docking onto their hosts, giant viruses, once dismissed as bacteria due to their size carrying hundreds of genes, enough to blur the line between the viral and the cellular. These structural forms are not just beautiful, they are functional. The shape of a capsid determines how a virus binds to a host cell. The surface proteins, called ligands, must match receptors on the host's membrane like a key fits a lock. If the match is wrong, the virus floats harmlessly. If it's right, the door opens. What happens next depends on the virus. Some inject their genetic material like a syringe. Others are engulfed whole, their envelope merging with the cell membrane to release their contents inside. Once in, the virus begins its takeover. If it's an RNA virus, it may be ready to start immediately. Some RNA viruses function as direct templates for protein synthesis, like orders handed straight to the ribosomes. Others carry their own enzymes to transcribe their RNA into a usable form. Retroviruses, such as HIV, go further still. They reverse transcribe their RNA into DNA and insert it directly into the host genome, becoming part of the cell's own code. DNA viruses, on the other hand, often enter the cell's nucleus. There they mimic the host's genes, co-opting the machinery to produce viral proteins and replicate their genome. In either case, the result is the same. The cell stops being itself. It becomes a factory, and once enough copies have been made, the virus escapes. Sometimes by sneaking out through exocytosis, sometimes by bursting the cell open, destroying it in the process. This is viral replication in its most basic form, but nothing about it is truly simple. Each step, from entry to assembly to exit, is a site of evolutionary pressure. Every virus is in a constant arms race with its host. Mutate too slowly, and the immune system catches up. Mutate too fast, and you lose function. It's a balancing act that plays out over billions of infections, across trillions of hosts every single day. This is where viral genetics get strange. Unlike most organisms, viruses do not pass down their genes in a neat vertical line, parent to child, generation to generation. They recombine, reassort, steal, drop, pick up. Some viruses, like influenza, have segmented genomes, meaning that if two different flu strains infect the same cell, they can swap segments like shuffled cards. This is how new flu pandemics arise, not from scratch, but from remix. Other viruses pick up host genes, sometimes accidentally, sometimes purposefully. Over evolutionary time, these stolen genes can be reinserted into other hosts, sometimes triggering cancer, sometimes changing regulation, sometimes doing nothing at all until, thousands of years later, we find them in our own genome and wonder, where did this come from? It came from the virus. It's here that we encounter paleovirology, the study of ancient viral sequences hidden within modern DNA. Using molecular clocks and phylogenetics, scientists can trace these insertions back through time. They can estimate when a particular retrovirus entered the human lineage. They can compare our genome to those of other mammals and see which viral fragments we share. Evidence of infections that occurred tens of millions of years ago. These endogenous retroviruses, ERVs, are fossils in a sense, not of bones, but of code. And like fossils, they tell stories. Some are benign, some are broken, some have been repurposed into human genes, active parts of our biology. One such example is Syncytin, a protein derived from an ancient viral envelope gene, now essential for placental development. Without it, mammals could not carry young internally. Think about that. A virus, once parasitic, now indispensable. What was once an infection is now inheritance. But how far back does this go? Could viruses have existed before cells? Could they be older than life as we know it? That brings us to a more speculative but fascinating frontier: viral origins. There are three main hypotheses about where viruses come from. The regression hypothesis suggests that viruses began as free-living cells that gradually lost complexity, becoming parasites. The escape hypothesis posits that viruses originated from bits of genetic material that escaped from cells, rogue segments that developed the ability to move between hosts. The virus first hypothesis takes it further still, suggesting that viruses predate modern cells, that they may have emerged from self-replicating molecules in the prebiotic world, relics of the RNA world hypothesis. This RNA world is a theoretical period in early Earth history when life was based not on DNA and proteins but on RNA alone, molecules capable of both storing information and catalyzing reactions. Many scientists believe that RNA came before DNA, that it was the first replicator, and if that's true, then viruses, with their RNA genomes and minimalist design, might be among the last surviving traces of that era. If so, viruses are not just ancient, they are primordial. They may be as old as life itself, and perhaps even older. Which brings us, gently, carefully, to astrobiology. If viruses can survive desiccation, freezing, radiation, if they can persist in permafrost for tens of thousands of years, if they can lie dormant, encoded within other organisms, then it's not unreasonable to ask, could viruses exist beyond Earth? Not necessarily our viruses, but viral-like entities, self-replicating genetic packages, in the subsurface oceans of Europa, or the methane lakes of Titan, in Martian soil, if life ever took root there, or in the interstellar dust that seeds new worlds. We do not know, but we are learning to look. NASA's Perseverance rover, exploring Mars, is not searching for viruses, is looking for biosignatures in rocks and regolith. But one day, we may send instruments capable of detecting viral analogues, protein shells, genetic fragments, replication patterns, because wherever there is life there may be something trying to copy it, to edit it, to ride it like a current. This is not fantasy, it is the logical extension of biology. If viruses are inevitable, if they emerge from the physics of replication, the mathematics of mutation, then we should expect them not as invaders, but as part of life's toolkit, an essential part. Even here, on Earth, we are still discovering new viromes, in deep-sea vents, in boiling acid springs, in Antarctic lakes beneath kilometers of ice. Each one adds new branches to the tree, or perhaps new roots hidden from view. And the more we look, the more we realize that the tree may never have had a single trunk. Life may not be a clean hierarchy, but a rhizome, a tangled web of horizontal exchanges, of coevolution, of viral whispering. We're only beginning to understand the grammar of that language. If you spend enough time with viruses, studying their structures, their genetics, their influence, you begin to realize something. They are not anomalies in the system. They are the system, not separate from life but folded into it like shadow into light. And nowhere is this more evident than in the immune system, the vast, quiet network inside every complex organism that lives in constant tension with viruses. A kind of molecular memory palace, built not just to repel invasion, but to remember it, study it, prepare for its return. Your immune system is ancient. Parts of it stretch back to the earliest multicellular life. Others are more recent, born from long exposure to viruses, shaped by the need to distinguish between the dangerous and the familiar. There are two main branches: the innate immune system, fast, broad, unspecific, and the adaptive immune system, slow, focused, precise. Viruses challenge both. The innate system acts first. It recognizes general patterns: double-stranded RNA, capsid proteins, unmethylated CpG motifs, molecular red flags common to many viruses. These are detected by toll-like receptors and other sentinels embedded in cell membranes and cytoplasm. The result is a cascade. Interferons are released, neighbouring cells are warned, and antiviral states are initiated. But this isn't always enough. Viruses are slippery. They mutate quickly. They hide within host cells. They mimic host molecules. Some even produce proteins that suppress immune responses, a kind of molecular camouflage. So the adaptive system steps in. T cells learn to recognize specific viral peptides. B cells produce antibodies tailored to unique service proteins. And once the infection is cleared, memory cells remain, ready should the virus return. This immunological memory is a kind of biological storytelling. It allows your body to remember battles it once fought, even decades later. And it allows us, through vaccines, to tell the body stories it hasn't lived through, to teach it about dangers it's never met, using fragments of viruses that pose no threat, but still trigger remembrance. Vaccines are among humanity's greatest inventions. They are blueprints of protection, and nearly all of them are built on viral understanding, whether through inactivated viruses, weakened ones, recombinant proteins, or genetic instructions that teach cells to make viral antigens themselves. This is where virology becomes a kind of peace treaty, a pact between understanding and survival. And yet, the war continues. New viruses emerge all the time, not just in human populations, but in ecosystems everywhere. Most never cross into people, many remain undiscovered, but as human activity expands into forests, wetlands, permafrost, the chances of viral spillover increase, and that brings us to a new field, viral ecology. It's the study of viruses not just as pathogens, but as ecological players, not killers, but curators. Because in the oceans, viruses are everywhere. A single milliliter of seawater contains tens of millions of them. They infect plankton, bacteria, archaea. They drive nutrient cycles. When a virus infects and lyses a marine microbe, it releases organic matter back into the water, fueling the microbial loop, sustaining the base of the food chain. This process is called the viral shunt, and it's massive. Without it, the oceans would be far less productive. Entire marine ecosystems depend on viral turnover, and in doing so, viruses indirectly regulate carbon flow, oxygen production, and even climate. Some viruses carry auxiliary metabolic genes, AMGs, that modify host metabolism during infection. These can boost photosynthesis in cyanobacteria, alter nitrogen fixation, or tweak sulfur pathways. They essentially rewire the cell temporarily, like a software patch, to optimize replication, and in doing so, influence planetary chemistry. It's not an exaggeration to say that viruses help regulate Earth's atmosphere. They don't do it with intent, they aren't conscious, but their cumulative activity scaled across the biosphere has real consequences, and this is why climate scientists are now paying attention to the virome. As permafrost thaws, ancient viral particles, preserved for thousands of years, may be released. Most are harmless, many are broken, but a few might still be viable. And even if they don't infect humans, they could interact with modern microbes in unpredictable ways, exchanging genes, altering balances, introducing forgotten elements back into the ecosystem. Climate change isn't just about temperature, it's about disturbance, and disturbance reshapes virospheres. Rising temperatures expand mosquito ranges, altering the geography of diseases like Zika, dengue, and West Nile. Deforestation compresses wildlife habitats, forcing species into closer contact and increasing cross-species transmission. Industrial agriculture concentrates genetically similar animals, creating ideal conditions for viral amplification and mutation. And on the microbial level, warming oceans affect the balance between microbial hosts and the viruses that regulate them. Shifts in this balance could alter oxygen production, greenhouse gas emissions, and nutrient cycling, all at planetary scale. So now we return to systems biology, the science of the whole. If you think of life as a network, genes, cells, organisms, ecosystems, then viruses are the signals passing between nodes. They're not just part of the network, they're what makes it dynamic. They facilitate gene flow, they create feedback loops, they introduce noise, and sometimes meaning. In synthetic biology, we now use viral elements to design gene circuits. In cancer therapy, we use viruses to selectively kill tumour cells. In neuroscience, viral vectors are used to trace brain pathways, infecting specific neurons with fluorescent markers, mapping consciousness one synapse at a time. Viruses are not just studied, they are harnessed, and yet we remain humbled by them. No model captures all their behaviour, no theory fully explains their origin, no algorithm predicts their next move with certainty. They are too small, too fast, too strange, and that is perhaps why they endure. Not just biologically, but philosophically. Because viruses remind us that life is not about control, it is about response, about complexity born from simplicity, about rules bent at the edges. They are the tiniest question marks at the end of existence. They don't speak, but they echo. And when we listen, truly listen, we learn not just about them, but about everything else. About what it means to change, to persist, to be part of a story that began before us and will continue long after. This is the science we carry now. It's in our vaccines, our climate models, our medical breakthroughs, our fears, our hopes, our metaphors. It is invisible, but not unknown. It is not soothing, always, but it can be. Because in the soft rhythm of understanding, there is peace. There is a kind of stillness, even in the complexity, the kind that lets you breathe easier, think slower, sleep maybe. And if you do, know that even as you drift, the world within you and around you is teeming with ancient quiet companions. Not good, not bad, just present, just about Earth. After the virus comes the quiet, not at first, not loudly, not like a door slamming shut or a film fading to black. The retreat of a virus is more like mist lifting from a valley, slowly, inconsistently, hesitant in places. It pulls back in stages, curling into corners, retreating from view long before it's truly gone. And what remains in its wake isn't silence, but space, emptied niches, changed ecosystems, new balances struggling to take form. This is the slow recovery. It happens after every outbreak, every spillover, every microscopic wildfire. The shape it takes depends on the scale of the event, the nature of the host, and the depth of the wound. Sometimes it takes weeks, sometimes centuries. Sometimes life doesn't return to what it was. It becomes something else entirely. Let's begin small. Imagine a forest, a bat roost hidden in its canopy, a virus, ancient, stable, harmless to its host, spills into another species, a primate perhaps. From the trees to the underbrush, from one troop to another, it spreads. Symptoms vary. Some suffer, some don't, some die, some carry it quietly, unaware. The virus burns through its available hosts. If it's well adapted, it might settle in, becoming endemic, a permanent, if subtle, part of the system. If it's too virulent, it might burn itself out. Either way, a ripple is made, populations shrink, territories shift, other species move into the space left behind, a predator loses its prey and starves, a plant once spread by fruit eaters stops travelling, grows only near its parent tree, slowly dwindles. A fungus blooms in the absence of grazing. This is how viruses rearrange ecosystems, not directly but consequentially, through chains of change, through dominoes that fall long after the first push, and while the system may settle, it doesn't settle back. There is no original equilibrium to return to. Nature doesn't keep backups, it only improvises. Now zoom out. Imagine a mass extinction, not caused by a virus, but punctuated by one, layered on top of global trauma like a fever atop frostbite. The Permian Triassic extinction, the Cretaceous Paleogene impact, the Devonian die-offs, the earlier, softer, slower collapses in the Precambrian Sea. In each, viruses may have acted as accelerants, finishing off weakened species, spreading in stressed populations, tipping the balance from survival to extinction. And then the slow rebuild. When an ecosystem collapses, its recovery isn't a reassembly, it's a reimagination. What comes back depends on what remains. After the Permian extinction, when over 90% of marine species vanished, it took at least 10 million years for biodiversity to fully recover. 10 million years of trial and error, of proto-crabs and gilled survivors, of empty shelves and sudden blooms. And among all of that, viruses were still there. They infected the new species. They rewrote DNA. They helped bacteria recycle nutrients. They acted, as they always do, not as directors but as catalysts, never planning the play, only rearranging the props. This too is a kind of recovery, and it's not just about loss. Sometimes the aftermath of a viral event includes invention. Consider the human placenta. Without it, mammals would not exist as we know them. And yet, part of what makes the placenta function, the ability of trophoblast cells to fuse and form a protective barrier, is driven by a protein that originated from an ancient retroviral envelope gene. A virus, long ago, inserted its DNA into a host genome. That gene stuck, was passed on, mutated, and eventually was repurposed into something essential. In this case, recovery was not just a return to health. It was evolution itself. Now shift again to the microbial world. When a bacterial population is devastated by phage infection, it doesn't just die. The survivors adapt. They develop resistance, sometimes through point mutations, sometimes through the acquisition of CRISPR arrays that remember the viral genome. These arrays are passed to daughter cells. The memory becomes collective, and over time, the population is no longer what it was. It is armed. It is changed. But viruses change too. They mutate to evade the CRISPR defense. They alter their entry points. They switch hosts. This dance of attack, defense, counterattack is not linear. It is circular, recursive, infinite. And so, recovery is never really an ending. It is a continuation, a movement into the next round. In human terms, we see this play out in our own histories. The 1918 flu pandemic reshaped not just public health, but how cities were designed. Ventilation, hospitals, school closures, community responses. It changed behaviour, hand washing, masks, distancing, in ways that echoed through generations. The HIV/AIDS epidemic redefined how science engages with affected communities. It accelerated the development of antiretrovirals. It changed sexual education. It exposed inequalities in healthcare. It mobilized global networks of activism, research and compassion. And COVID-19, still echoing in memory, redefined everything from supply chains to mental health to our understanding of indoor air. Long COVID, a lingering echo in the body, has expanded our view of what recovery even means. Because sometimes recovery isn't restoration, it's adaptation. It's learning to live with something rather than without it. And viruses are often permanent. Some remain latent, hiding in our nerves, our DNA, our populations. Herpes viruses, for example, live in a state of uneasy truce with their hosts, occasionally reactivated, often asymptomatic. Part of the background noise of life. Others shape populations without individual symptoms. Cytomegalovirus infects most people silently, but its presence influences the immune system, shifts T-cell distributions, may even affect aging. In this way, recovery can be invisible, not marked by a finish line but by a settling, a reframing, a new normal. And in ecosystems the same is true. A forest regrows after disease, but the species mix is different. The soil chemistry shifts, the insect life alters, the predators adapt. A coral reef hit by bleaching and viral outbreaks may regrow, but with hardier species, new symbiotic relationships, altered structure. The biosphere learns, not consciously, not with intention, but through persistence. And in every environment the viruses remain, not just as threats, but as archivists. They carry genes between hosts. They facilitate gene exchange across species. They help keep microbial populations in check. In soil, they regulate bacteria that fix nitrogen, influencing plant health. In water, they determine which phytoplankton dominate, influencing global oxygen levels. In animals, they modulate gut microbiota, influencing digestion, immunity, mood. And in humans, they quietly pass from hand to surface to breath. Sometimes harmful, sometimes harmless, sometimes helpful. The human virome, the collection of all viruses in and on our body, is still largely unmapped. We know it's vast. We know it varies. We know it shifts over time. And we suspect it plays a role in health and disease far beyond what we currently understand. Recovery, then, is not just about surviving a viral encounter. It is about integrating it. Because in a way, we are part virus. We carry their genes. We respond to their patterns. We build our defenses based on their shapes, and over evolutionary time they have become part of our design. So when we speak of recovery, we must broaden the term. It is not a return to stasis. It is the continuation of a dialogue between host and parasite, between past and present, between destruction and creation. This is the rhythm of life on Earth. Disruption, collapse, emergence, integration, over and over, in the fossil record, in the genome, in the lungs of a single bat, hanging upside down in a silent tree. And in that sense, viruses are not just agents of disease, they are agents of change. They prune, they push, they provoke, and what follows them is not always devastation. Sometimes it is transformation. Sometimes the world they leave behind is not broken, just different, more resilient, more complex, more aware of itself. And so the slow recovery continues, in forests, in cities, in tide pools and genomes and quiet bodies asleep beneath blankets of air. You may not see it, but it's happening, right now, all around you. So much of what viruses do, we don't see. They arrive invisibly, multiply silently, move through the air, across the skin, into bloodstreams and breath, with no footsteps, no sound. And yet, they leave behind reshaped forests, rewritten genomes, reimagined futures. And it makes you wonder, what else in this world shapes us quietly? Because viruses at their heart are stories. They carry information, they pass it on, they change their hosts. That's what stories do. They survive by replication, by resonance, and they adapt with every telling. We often imagine viruses as intruders, disruptors, villains. But what if they're more like punctuation marks? What if they're how life changes the subject, takes a pause, shifts the sentence, starts a new paragraph? Every so often, Earth has been interrupted by fire, by ice, by impact, by oxygen, and perhaps by virus. Interruptions don't always destroy. Sometimes they clear space for something new. When the oxygenation event wiped out anaerobic life, it also made room for the explosion of multicellular organisms. When a virus wipes out one species, another may rise to fill the gap. When cells are infected, genes are shuffled, rewritten, occasionally even improved. So yes, viruses are dangerous, but they're also transformative. They're evolution's accelerators, editors, shadows. They pull things apart, and in doing so, open space for the unexpected. And that makes them a mirror for our times, because we too are living in an age of acceleration, of compression, of events piling atop one another like waves against a breakwater. The climate is changing, ecosystems are collapsing, viruses are crossing species boundaries, continents, oceans, faster than ever before. But so are ideas, so are warnings, so are acts of care. In this moment, when the line between crisis and change has never been thinner, viruses remind us that disruption is only one part of the story. What comes after, how we respond, how we recover, what we choose to become, that's the rest. Look back just a few years. A virus reshaped the world. COVID-19 froze cities, silenced skies, closed borders, redefined normal. It reminded us how fragile modern life is, how fast things can fall apart, how deeply interconnected we truly are. It also reminded us how fast science can move when it must. Vaccines developed in under a year, genome sequencing done in days, global collaboration at a scale never seen before. Yes, there were failures. Yes, there was grief. But there was also astonishing adaptation. And in that lies a question. What else could we respond to if we chose to? The climate, mass extinction, antimicrobial resistance. Viruses aren't the only silent forces shaping our world. Carbon too is invisible. So is habitat loss. So is ocean acidification. And yet their effects, like those of a virus, ripple out in cascading chains of consequence. The difference is that viruses don't wait for permission, but humans do. We wait to be certain. We wait to be safe. We wait to be asked. And maybe that's the deeper lesson here. Viruses remind us that the world doesn't pause until we're ready, that sometimes the old systems have to be interrupted before anything new can emerge. And maybe, just maybe, the interruption is already here. So what now? If viruses are one of nature's most powerful forces of change, what does it mean that we are now the most powerful force on Earth? That we move more nitrogen than all the world's rivers? That we've cut, dug, burned, reshaped more of the biosphere than any ice age or asteroid? Are we the virus now? It's a tempting metaphor, and a dangerous one. Because viruses aren't evil. They aren't invaders. They're part of the system. And so are we. We are not outside of nature. We are not above it. We are expressions of it. Complex, reflective, sometimes destructive, sometimes healing. Like a virus, we replicate, we spread. But unlike viruses, we choose, we reflect, we mourn, we hope. And that capacity to see our impact and alter it may be what allows us to become something more than just another acceleration. Maybe like a good virus, we can leave behind useful code. Maybe our legacy won't just be carbon and concrete, but knowledge, conservation, healing. Maybe we can engineer resilience instead of fragility, regrowth instead of ruin. Because we are not helpless. Every ecosystem we've damaged has shown some ability to recover, given time, space, protection. Coral reefs can regrow, forests can rebound, populations can return, and so can we. But recovery starts with perspective, with remembering that we are not the main character in Earth's story, We are a subplot, a recent chapter, a single thread in a tapestry 4 billion years long. Viruses remind us of that too. They have been here since the beginning, and they will likely be here long after us. And somehow that's comforting, because it means life goes on, not always in the form we expect, not always in the direction we hoped, but it goes. Viruses are not here to end the world. They are part of what makes it move, and so too are we. So we ask, how do we move with care? How do we disrupt the right things? How do we become a force of recovery, not collapse? It starts perhaps with humility, with understanding that survival isn't dominance, it's adaptability, it's balance. It's remembering that even our immune system, that miraculous shield we all carry, is shaped by millions of years of peaceful coexistence with the microbial world. We don't just fight viruses, we live with them. We are, in part, made of them. So maybe, instead of fearing every invisible thing, we begin to listen to them. What is the virus teaching? What is it revealing? What have we forgotten? Perhaps it's showing us how much we need each other, how health is collective, how one person's breath connects to another's lungs, how one forest's collapse can trigger disease in a city halfway around the world. Perhaps it's reminding us of scale. That change doesn't have to be loud to be real, that the smallest things, a mutation, a cough, a choice, can ripple outward. And perhaps most of all it's asking us to pay attention, because the systems that viruses move through, blood, soil, atmosphere, are the same systems we move through. When we harm them, we harm ourselves. When we heal them, we heal something larger. So here we are, a species shaped by viruses, now shaping the planet in return. What we do next matters, because we're not just reacting to the past, we're writing the next chapter. We can choose to build immunity into our systems, not just biologically, but socially, ecologically, politically. We can choose to invest in care over conquest, cooperation over control, complexity over simplicity. And we can choose to be a kind of beneficial virus, to carry forward something helpful, to insert ourselves gently into the future, to replicate not just ideas but compassion, to spread not just technology but wisdom. Because in the end, life on Earth is not a war. It is a conversation, long unfinished, filled with interruptions and pauses and changes of subject. And viruses have always been part of that grammar. So have we. And now, perhaps we listen more closely to the virome, to the biosphere, to each other, to this strange shared story of being alive together on a planet that is itself a kind of living system, fragile, adaptive, still learning, still recovering, still full of wonder, still just about Earth. And so we end, not with a final answer, but with a widening question. Where did the first virus come from? We may never know. Not entirely. Not in the way we know the age of a rock or the orbit of a moon. Viruses leave no bones, no voice, no lasting trace that isn't buried deep in the folds of other lives. But what we do know is this. They've always been here. They've shaped us, challenged us, rewritten us. And somehow, they still remain. Quiet companions, invisible teachers, forever changing and being changed. Maybe that's enough for now. Maybe tonight it's enough to feel the shape of the question. And let the answer be a kind of wonder. If you've made it this far, listening, drifting, thinking, thank you, truly.